Imagine a world without antibiotics, without vaccines, without treatment for countless diseases. Every single one of these life-changing innovations started with a simple question, a spark of curiosity, and meticulous investigation. Welcome to the invisible yet immensely necessary world of basic research. In the vast and complex journey of drug development that we briefly reviewed last time, this is our critical first step. It's where the magic of discovery truly begins, the origin story of new therapies that eventually become life-saving medicines. Think of it as the laying the foundation of a skyscraper. Without a strong foundation, nothing else can be built on it. Our journey begins with what I would call identity stage. This is where academic and industry scientists collaborate, driven by a shared mission, which is to find the root biological malfunction that causes a disease. When I was taking a med school class, the professor at the time said, "Your future patient is a cell." This didn't make sense at the time, but this means to truly understand a disorder, you must trace it back to a single cell that has gone haywire. More specifically, a single protein or a gene within that cell that is often the primary culprit. Let's think of it like a story. Once upon a time, a young scientist named Dr. Hadi was studying a rare genetic disease. Patient with condition had a specific protein that was misbehaving, causing a chain of reaction of cellular problems. Most people saw a confusing web of symptoms, but not our young scientist who saw a culprit. He and his team spent years meticulously mapping the protein's function and structure, looking for its unique functions. Their work wasn't about creating a drug yet. It was about understanding the link for the underlying malfunction at its core. This deep dive into a disease at the molecular level, looking for genes, proteins, or signaling pathways that are part of a problem, is the first and most critical step. It's a lot like being a detective, tracing complex crime back to a single person. Once that target is identified, we move to the fix-it stage. We now turn our focus on discovering and refining a therapeutic modality that can either modulate or permanently correct the function of our identified target. Let's continue our young scientist's story. Now that they have identified and know the misbehaving protein was this target, the team had a clear goal. The first step was a massive screening effort where they tested thousands, sometimes millions of different small molecules against the target to find the one that showed any promising activity. This is like sifting through a gigantic pile of sand to find a few grains of gold. Now, a critical point here is that the approach depends entirely on the type of therapy being pursued. Screening for a small molecule drug is a very different process from approaching it with large molecule therapeutic protein or gene therapy. We wouldn't go into the details of these differences at this point, but just know that they take different routes. Once they had the initial hit or lead, the next phase was optimization. Here the team had to work like master craftspeople, tweaking and refining these initial candidates to make them as effective and precise as possible. And finally, we reach the prove it stage. Here we validate our optimized therapeutic candidates. We perform rigorous tests in the laboratory, primarily using in vitro models or in test tube models, and with small animal models. To complete our young scientist's story, his team had found a promising new therapy. They first tested it on cells in a petri dish that had the same genetic mutation as the patient. They watched as the new therapy successfully restored normal function. This was their in vitro proof. Next, they moved to a controlled experiment using small animal models with the same disease. They administered the therapy and observed it had the desired effect on the animal's health without causing any major side effects. These initial tests proved that the crucial proof of concept. To complete our young scientist's story, his team had found a promising new therapy. They first tested it on cells in a petri dish that had the same genetic mutations as the patient. They watched as the new therapy successfully restored normal function. This was their in vitro proof. Next, they moved to a controlled experiment using small animal model with the same disease. They administered the therapy and observed that it had the desired effect on the animal's health without causing any major side effects. These initial tests provided the crucial proof of concept, confirming that their work had led to a truly promising therapeutic candidate. And more broadly, the impact of basic research extends far beyond these 3 steps that I described. The applications are vast, from cutting-edge techniques like gene editing and 3D organoids, which are miniature lab-grown organs, to detailed studies of proteins expression, signaling pathway, and structure. Basic research provides the tools and understanding that fuels the entire drug development pipeline. The knowledge gained from these fundamental exploration is published, shared, and built upon by scientists all over the world. It's a continuous cycle of discovery where new knowledge begets new questions, paving the way for next generation of breakthroughs. So, the next time you hear about a new drug or treatment, remember the tireless work happening in labs worldwide. It is these silent heroes and their fundamental quest for knowledge that ultimately unlocks the cures of tomorrow. If you found this video to be insightful, please like the video, subscribe for more videos, and let me know in the comment box what other topics you like to explore next. Until next time, adieu.